And the replication of process of DNA, it suggests that two strands of DNA, they separate and they act as templates. 50% of the strands will have only the light strands, 50% of the strand will have both the light and the heavy strands. Both the strands, they unwind and then they participate in the process of replication. We call it a semi-conservative method of replication. Hello everyone, a warm welcome to this session for class 12 biology. I am Dr. Divya, biology faculty. So in this session of chapter 5, that is molecular basis of inheritance, we will learn about the replication process in DNA, wherein the replication is a semi-conservative method, and what experiments were done to prove that DNA replication is semi-conservative. We will learn about that. So talking about replication, the word itself says replication, meaning production of replica of something. Replica means copies, the same exact copies, or we can also call them the clone, right? So here the DNA, multiple copies of the DNA needs to be produced. Why DNA should produce multiple copies? When during the process of mitosis or meiosis, when one cell needs to divide into two daughter cells, then whatever genetic material is present here should get equally distributed, having the same gene in them to the daughter cells also. Say for example, we are multicellular. We have numerous cells in our body, right? And how did we get that numerous cell? We were a single cell earlier, meaning in the mother's womb, we were a single-celled zygote, diploid zygote. That is, our birth started from, or our generation started from being a diploid unicellular zygote to multicellular organism, the way we are today, right? So here, if n number of such cells need to be formed, each cell, the cells need to divide and multiply. So when they divide and multiply, the chromosomes should get equally distributed into daughter cells. So when the chromosomes get equally distributed, along with that, the DNA also gets distributed. Now, DNA, if it needs to get distributed, copies of it needs, it's just like taking a Xerox of one sheet of paper, right? So the same thing, same question paper, everybody, you will get. Why? Because copies of the same question paper is being created. Same way here also, copies of the same strand of DNA need to be created. If copies of the same strand are not made, then how will the daughter cell get the same genes from that parent cell also? So that is why that process is called as replication, or making copies of DNA or generating copies of DNA. We call it as replication. And this mechanism of replication was first proposed by Watson and Crick. And we know that Watson and Crick were the first person to discover DNA, that is the double helical structure of DNA. So DNA is made up of nitrogenous bases along with a pentose sugar, along with, that is a deoxyribose pentose sugar, along with phosphorus backbone. All that idea was given by Watson and Crick. In the same way, they also proposed the replication process of DNA. And the replication of process of DNA, it suggests that two strands of DNA, they separate and they act as templates. So now we have a DNA strand. It's the same thing that I'm drawing here. So DNA strand which are coiled. Now, to make copies of it, we need, it's just like the teacher prepares a question paper and takes it to the Xerox machine, puts it in the Xerox machine, and she'll give number of copies, she'll just give there, and the Xerox machine will give the same question paper and number of copies, right? So, without that question paper being prepared by the teacher, one blank sheet, if we go and keep it, give it for Xerox, now will the Xerox machine prepare a question paper? No. It already needs a prepared question paper which it can take copies of. The same way here, already existing strands in the DNA are the ones which participate in the process of replication. And those strands we call as the template strand. And we know that in DNA, there are two strands that are present, and hence it is a double-stranded DNA, right? So this one of the strands which runs from the 5' direction to the 3' direction, and the other one from the 3' to the 5' direction. So this is one of the template strand, and this is one of the template strands. So how many template strands are there? Two template strands, or we can also call them as the parental strands. So the strands which participate in the process of replication, they act as a template or they act as a copy for the synthesis of new complementary strand. So for the formation, so this in the next session we'll study how exactly replication happens, what are the machinery or what are the enzymes that are involved in the process of replication and all that. Now these strands, now we'll just, I'll give you an idea of what replication is. So here these strands, because I'll just give you an idea of what replication is, because here we are concentrating only on semi-conservative mode of replication in an experiment that was done to prove it. So that is why. So these template strands at certain region, they need to separate. They will separate like this, because if they remain coiled, there is no space for the coding strand, for the complementary strand to get formed, right? So they need to separate. At one point on the DNA helix or on the DNA strand, they will separate or they will unwind. I can use the word unwind. The DNA which was coiled at certain region or at certain sequence, they will just unwind. Now we have two separate unwound strands or two separate templates. Now for this complementary strand needs to get produced. So this, now how many did we get? From 1 we got another 1 and 2. 2 copies of DNA. Now these 2 copies will again participate in the process of replication to form 4 copies, then 8 copies, then 16 copies. That is how DNA keeps on replicating or multiplying in number. So for this complementary strands means if here A is there, then here it will be T. If here T is there, it will be A, right? So because I told in the previous session, adenine complements with thymine, guanine always complements with cytosine, and vice versa, like that. So therefore this is how the DNA replication happens. In detail we'll study in the next session. So after completion of replication, each DNA molecule now will have one parental strand and one newly synthesized strand, right? So this will be the parental strand, or which is called as the template strand. I'll write it as P. This will be the parental strand, and these two will be the newly synthesized strand. I'm writing it as NS. So newly synthesized strand. So there will be a parent strand and there will be a daughter strand. So that is how DNA replication happens. And because both the strands, they unwind and then they participate in the process of replication, we call it a semi-conservative method of replication. Why? Because they are using some of the conserved nitrogenous bases that are present on the parental strand to create a new strand complementing to it. So that is why DNA replication is semi-conservative. Now, if we just say to the people DNA replication is semi-conservative, will the people believe? No, you need experimental proofs to prove that, right? So for that, Matthew Messelson and Franklin Stahl, they conducted an experiment to prove that the mode of replication in DNA is semi-conservative method. So they proved that DNA replicates semi-conservatively, and the first organism that they choose for studying the semi-conservative mode of replication was in the bacteria, that is Escherichia coli. So these two are the scientists, Matthew Messelson and Franklin Stahl, who contributed towards the experiment to prove that DNA replication is semi-conservative. So let's look into what exactly did they do in their experiment. So first I told you they choose the bacteria E. coli. Why they choose E. coli is they don't do studies on humans, because human cells, the growth rates are quite slow. But bacteria, they tend to multiply very fast in a very fast rate. So that is why it is easy to finish off the experiments and get results within few minutes itself. So that is why. So first what they did is they took the E. coli bacteria from their culture plate, that is Escherichia coli bacteria from their culture plate, and they put it into a medium. So for your easy understanding, I'll draw a Petri plate. So Petri plate, they are just like small plates or saucers wherein it will have a lid on that, and in that Petri plate can be made up of glass or plastic. So in that we pour some media, because anything you need to grow, you need to have a media, artificial media. So for bacteria, we have a bacterial medium that is used. So here they used a media, bacterial media they put in that, and along with that bacteria media, they put a chemical, that is 15NH4Cl. So what did they put here? 15NH4Cl. Cl. That is ammonia chloride. They used a chemical that is ammonia chloride. What type of ammonia chloride? Ammonia chloride in which its nitrogen has 15. That is, the nitrogen has a 15N isotope. So 15N isotope means it will be a heavy. The chemical will be heavy. So that is what they added. That is 15N is the heavy isotope of nitrogen. So in one media, now in that they grew this bacteria. Which was that bacteria? E. coli. This is, they put the media into the Petri plate. Media containing what? Ammonia chloride, which is having a 15N isotope. So nitrogen's isotope is 15N. And they waited for few minutes of hours, and the result was that 15N got incorporated into the newly synthesized DNA. Now when these bacteria, maybe they had around 5 bacteria they introduced into the culture plates. Within few minutes it will multiply to 20 or 15 or like that. It will multiply, right? So when they are multiplying, obviously new strands of DNA need to get formed. So new strands of DNA, how it will get formed? Same way, by the process of replication, like how I said earlier, right? So when they checked it, they found out that in the newly synthesized strand, 15N isotopes was also there. It got incorporated, 15N isotope got incorporated in the DNA strand. So now they have a DNA. Which one? From this culture, they got a DNA which is having 15N isotope. So I'll write it as 15N DNA. In which it is there? It is there in the E. coli bacteria now. So the E. coli bacteria, which was cultured in a media containing ammonia chloride, which is having a heavy isotope of 15N. After few minutes of multiplication of the bacteria, they observed that the DNA in the newly synthesized or new cultures of bacteria which had formed here, they contained DNA strands in which 15N or the heavy isotope of nitrogen 15N was present. So this is now what they have in the culture plate. This heavy DNA molecule can be distinguished from the normal DNA molecule by centrifugation in cesium chloride. Now, along with that, they did one more experiment. So, I'll go to that later. So, here what they did was, now they wanted to separate it. So, for separation of the DNA strand, there is a technique which is used, which is called a centrifugation technique. In centrifugation technique, there will be centrifugation vials that are there. So, the vials will be almost like this. So, in this vial, they'll add some solution. So, in this case, they added the chemical cesium chloride. Now, why specifically did they add cesium chloride? Because cesium chloride is one such chemical that can be used in the separation process in centrifugation, and especially that is being used because cesium chloride has the capability of extracting and separating DNA strands from the RNA and from the proteins and all that. Because in all the DNA strand, they will have an RNA strand also, because mRNA will be participating, right? So that is why. So that this chemical has that capability. So that is why it is used as a centrifugation solution. Now what did they do? They took this E. coli, which is having 15N DNA, that is heavy isotope DNA, and they put it in the centrifugation tube which is having cesium chloride, and they put that tube into the centrifugation machine. Centrifugation machine just looks like a vessel wherein lots of tubes can be kept, and we can set the rotations like 1000 RPM, 10,000 RPM, 2000 RPM, like any RPM suggested by the scientist, whoever is doing the experiment. So that RPM, when it turns or rotates the centrifugation tube inside, and it will be subjected to rotation. At that time, what happens? The strands will get separated based on their heaviness. Now, all the DNA strands will have the same weight. So that is why we can't separate the strands easily. So that is why what these two people, scientists, did was they used 15N, that is they used ammonia chloride in the media having a heavy isotope of 15N in it. So therefore, now when 15N got incorporated in the DNA, didn't the DNA become heavy? Now when we have heavy DNA, light DNA and all that, it is easy to separate by the process of centrifugation. So that is why. So moving further, we will just look at this and see exactly what did they do. So I had already told you So we got to know that from the previous slide, E. coli was grown. To that culture media itself, they added ammonia chloride, in which it contained a heavy isotope of nitrogen, that is 15N. Now, in those E. coli bacterial DNA, these 15N got incorporated. Now, the E. coli bacteria is having what kind of DNA? A heavy DNA, which is having a 15N isotope, right? That what they did was they put it into centrifugation force. Why did they put it into centrifugation force? Because to separate the DNA strand. Now, the DNA is double helix. Inside the organism, it will get separated and all that because of some of the enzymes that are taking place inside the E. coli. But when we take out the DNA strand, we can't add those enzymes, right? So that is why they separated. So for separation, what did they use? They used the chemical cesium chloride. So when it was subjected to centrifugation, the DNA strands got separated in the centrifugation tube. See, like this, they got separated. And why is it at the bottom? It's not floating at the top. Because it is having, the DNA strand is having what? It is having the heavy isotope 15N in it. So that is why it is at the bottom. It's not floating at the top. So this is how it got during centrifugation, they got separated. So to make you easily understand, these are the two DNA strands. For example, I'll draw it like this. One of the strand is having 15N DNA and the other strand is also having 15N DNA. And they were put into the centrifugation tube containing what? Cesium chloride. And they were put for rotation. Once the rotation happened, the strands got separated and both the strands got settled at the bottom of the centrifugation tube because both the strands are heavy. So that is why. Now this is clear. So next what happened? They removed, again they put this to centrifugation. So what they did was, now they removed this bacteria, they incorporated this DNA into the bacteria again, and those bacteria, they grew it in culture media. Now in that culture media, what did they add? So I'll just draw it here. They grew it in the culture media. Which one? These bacteria. These bacteria which are having 15N DNA in them. E. coli bacteria having 15N DNA. Now in this culture media, in the first culture media, what had they added? They had added only 15NH4Cl, right? In this culture media, they added 15NH4Cl along with 14NH4Cl. Now therefore, what is there? We are having one heavy strand and there is one light strand, because some of the bacteria, they took up 15NH4Cl and they took up both, that is the DNA strands in the bacteria. They took up the heavy isotope 15N and also the light isotope. So to make it clear, I'll just draw it like this. So now in the bacterial DNA, what is there? One of the strand will be having 15N DNA. The other is 14N DNA. So that is how it is. Now, the work of what Messelson and Stahl did, again they put it to centrifugation. So they put that, they took this E. coli and they they separated the DNA strands, they isolated the DNA and they put it into centrifugation. Now this is the generation 1, right? So in generation 1, when they subjected it to centrifugation, again where in cesium chloride, they found out that the strands that had got separated, one was lighter in weight, therefore it was floating at the top, and at the other one is heavier in weight and therefore it is floating at the bottom. So you can see here, one of the strand is having 15N DNA and the other strand is having 14N DNA. Now, this 15N DNA and 14N DNA, in this, which is the parental strand, the parental strand is, one of the parental strand will be the 15N DNA, and for that a newly synthesized strand will get formed of the 14N DNA. So that is what they found out. Now again after 40 minutes, that is again they put this into, again they cultured it for 40 minutes and again they put it for centrifugation. Now here these two strands, they subjected it to centrifugation. So when it got separated, what happened? In generation 2, there was one whole set of DNA that was produced and both the strands were 14N DNA and another set of DNA, because from these two DNA strands we can get four strands, right, altogether. That is 4 parental strand and 4 daughter strand. Therefore, 4 DNA molecules we can get here. So here one of the DNA strand had 15N DNA and one more had 14N DNA and the other one had completely 14N DNA. So therefore, 50% of the strands will have only the light strands. 50% of the strand will have both the light and the heavy strand. So therefore, they got to know that only one strand of the DNA participates in the process of replication here, and therefore they got to know that the DNA replication is semi-conservative in nature. So this was their experiment to prove that out of the two strands, the strands unwind and only one strand, that is the strands unwind and each strand participate in the process of replication, forming two daughter strands and therefore 2 DNA molecules in generation 1 and 4 DNA molecules in generation 2. Now this was for 40 minutes. So if it is subjected to 80 minutes, more number of lighter strands will get produced, that is about and less number of heavy strands will get produced, that is somewhere about around 12.5% of heavy strands will get produced and rest of the strands, that is somewhere around 87.5, somewhere around 87.5% will be the hair, that is lighter strands. So it's somewhere around 7:1 ratio it will be. So this proves that as and when we are culturing the E. coli for further minutes, that is for about 80 minutes, then there will be variation in the number of heavy strands and the number of light strands that will get formed. So this is about, that is hybrid DNA. So this we can call it as hybrid DNA, both of these. These two are hybrid DNAs. Why? Because one of the strand is heavy strand and the other strand is light strand. It's usually this experiment was just done to show that only one strand participates in the process of replication. So this was about this session wherein we learned that DNA replication is semi-conservative with the help of Messelson and Stahl's experimental proof to state that yes, DNA replication is semi-conservative wherein one strand participates in the process of, the strand unwinds and each strand participates in the process of replication, wherein one DNA molecule will form 2 daughter DNA molecules and 2 will form 4 and therefore it will go on increasing like that. So in the next coming session, we will learn about mechanism of DNA replication, how exactly DNA replication takes place. As I told you, the coil, that is the DNA strand, they need to unwind at the place, right? That is origin of replication. There only they have to unwind. They can't unwind anywhere else. So how exactly that happens, and for the replication to happen, and a number of enzymes are involved. So what are those enzymes? What is the mechanism of those enzymes? We shall look into. So see you in the next coming session. Thank you.